In this video, we will look at the process of binary fission, bacterial growth using mean division time, how microorganisms are cultured in laboratories, and finally, we'll see how scientists test the effectiveness of antibiotics. So, what exactly is binary fission? Well, the name gives us a clue. Binary means two, and fission means splitting. So binary fission occurs when a single bacterium divides, creating two genetically identical daughter cells. But there's more to this process than just simple splitting. Let's break it down step by step. First, the circular DNA and any plasmids within the bacterial cell replicate. As this happens, the cell grows larger to accommodate the newly duplicated genetic material. The replicated circular DNA strands then move to opposite ends or poles of the cell. Next, the cytoplasm begins to divide and new cell walls start forming. Finally, the cytoplasm completely divides, resulting in two daughter cells. Each daughter cell has one copy of the circular DNA, though they can have different numbers of plasmid copies. What's truly remarkable about binary fission is the speed at which it can occur. When environmental conditions are optimal with sufficient nutritional resources and appropriate temperature ranges, certain bacterial species, including E. coli, can undergo a complete cell division cycle in approximately 20 minutes. Just imagine 100 bacterial cells could become 200 in 20 minutes, then 400 in 40 minutes, then 800 in an hour. This is why bacterial infections can develop so rapidly in the body. However, if conditions become unfavorable, perhaps due to lack of nutrients, extreme temperatures, or the presence of antibiotics, bacteria will slow or stop dividing and may eventually die. Because bacteria multiply at an exponential rate, scientists use mathematical formulas to predict population size. If you know the mean division time of a bacterial cell, you can calculate how many cells will be present after a certain period. Let me show you how this works with an example. Say we have a bacterial cell with a mean division time of 45 minutes, and we want to know how many cells it will produce after 3 hours. First, we convert all our time measurements to the same units. 3 hours are switched to 180 minutes. Next, we divide the total time by the mean division time to find the number of divisions. 180 minutes divided by 45 minutes equals 4 divisions. Finally, we calculate 2 raised to the power of the number of divisions, so 2 to the power of 4 equals 16 cells. So after 3 hours, our single bacterial cell would have multiplied to become 16 cells. Now, let's move on to how scientists grow bacteria in laboratory settings. This process is called culturing, and it's essential for studying bacterial characteristics and testing antibiotic effectiveness. To culture bacteria, scientists need to provide them with all the nutrients they require. This is done using a culture medium, a substance containing carbohydrates, minerals, proteins, and vitamins necessary for bacterial growth. Scientists typically use two distinct types of growth media, either liquid solutions called nutrient broths or solidified media containing agar. Agar is particularly useful because bacteria grown on it form visible colonies that scientists can observe and count. First, hot liquid agar is poured into shallow plastic dishes called petri dishes and allowed to cool and solidify. Once the agar has set, bacteria can be transferred to it using sterilized tools like inoculating loops. These are small wire loops that can pick up tiny amounts of bacteria. Alternatively, sterile droppers and spreaders can be used to create an even layer of bacteria across the plate. For safety purposes in educational settings, microorganism cultures are maintained at temperatures of 25 degrees Celsius or below, since dangerous pathogens tend to proliferate more readily at higher temperatures. However, in industrial settings where safety measures are more robust, cultures might be incubated at higher temperatures to accelerate growth. One of the most important applications of bacterial culturing is testing the effectiveness of antibiotics. This helps scientists discover new antibiotics and determine in which ones work best against specific bacterial strains. Here's how this investigation typically works. First, an agar plate is prepared with an even covering of bacteria spread across its surface. Next, small paper discs are soaked in different antibiotics or different concentrations of the same antibiotic. These discs are placed on the agar surface, spaced apart from each other. As the antibiotics diffuse into the agar, they will kill bacteria that are sensitive to them. After incubating the plate for about 48 hours at 25 degrees Celsius, clear zones called inhibition zones appear around the discs where bacteria have been killed. The diameter of these clear inhibition areas relates directly to antibiotic potency. More effective antibiotics produce larger zones where bacterial growth is prevented. A control disc soaked in sterile water rather than antibiotic is always included. This ensures that any observed inhibition is due to the antibiotic itself and not some other factor. When working with microorganisms, preventing contamination is crucial. Unwanted microorganisms can ruin experiments and potentially cause harm. Here are three key steps scientists take to maintain uncontaminated cultures. First, they sterilize inoculating loops by passing them through a flame before transferring bacteria. Additionally, they will seal petri dishes with tape to prevent airborne microorganisms from entering. And finally, they will store these petri dishes upside down to prevent potentially contaminated condensation from dripping onto the bacteria. These precautions ensure that the only microorganisms growing in the culture are the ones being studied. Binary fission and bacterial culturing are fundamental concepts in microbiology with widespread applications in medicine, research, and industry. Understanding these processes helps scientists combat disease and advance our knowledge of these microscopic organisms.